Welcome, everyone. I would like to start by acknowledging the traditional owners and custodians of the land on which we are meeting today, the Wurundjeri people of the Kulin Nation, and pay my respect to their elders past and present. It's a really great pleasure to welcome our today's seminar speaker, Professor Sharon Lewin. Sharon is an infectious disease physician, but also a highly regarded basic scientist. She's the director of the Peter Doherty Institute of Infection and Immunity across the road. And she's an internationally renowned and one of the world leading and influential scientists with focus on HIV, and in particular HIV latency and persistence. She has published over 260 papers, many of them in the top tier journals, and Sharon is a member of the NHMRC Council and an elected member of the Governing Council of the International AIDS Society, IAS, representing the Asia-Pacific region. Sharon was named Melburnian of the year 2014, and in 2015 she was awarded the highly prestigious Peter Wills Medal from Research Australia. And she's actually the first woman who have received this award. It's a great pleasure to welcome you, Sharon, today. Sharon will give us an introduction, what we know about HIV latency and current strategies to eliminate latency and ultimately cure HIV. Thanks, Sharon.
Thank you very much for the kind introduction, and great to see so many people here interested in infection and immunity. So I'm going to assume that most of you don't know a lot about HIV and give you quite a good background to understand why we're looking for a cure, and then move into a few interesting areas of research that my own group is focusing on. So for those of you that don't know a lot about HIV, I hope you know that there's been tremendous advances in the treatment of HIV. So on the right is literally what people take to control HIV currently, a single tablet, co-formulated with 2 or 3 drugs. And if individuals take their treatment every day, viral load remains under control. Treatment is cheap. You can now treat people with this sort of antiviral strategy for $100 a year, and about 50% of the world's people living with HIV now have access to antiviral therapy. So an incredible, spectacular advance in global health, often cited as one of the biggest medical advances in the last 3 decades. And I've had the great privilege, actually, of working in HIV since really the very beginning. I started I was a medical student in 1981, the first cases of AIDS being reported, HIV being discovered in 1983, and then to have seen these tremendous advances, actually not just in treatment, but in prevention, has been quite extraordinary. So when you put people on antiviral therapy, you can very accurately measure the amount of virus in the blood. We call that someone's viral load. We can literally calculate copies of HIV RNA per mil of blood. And most people will have, tens of thousands, hundreds of thousands, millions of copies per mil of HIV RNA per mil in untreated HIV infection. And as soon as people start treatment, our viral load pretty much reduces down to what we call undetectable levels within 4 weeks. And undetectable just means that the commercial assays no longer can detect virus. Most commercial assays detect down to about 20 copies per mil. And people can lead very normal, happy lives. Life expectancy returns to normal, and in fact can no longer transmit HIV sexually, an incredible advance that we only really proved in the last 2 to 3 years. But as soon as people stop treatment, pretty much between 2 to 3 weeks after stopping treatment, viral load rapidly rebounds. So we know that although the viral load is undetectable at this point for many, many years, virus is still present. And actually, if you had a very sensitive assay, and we do have these sensitive assays in the laboratory that measure down to 1 copy per mil, you can pretty much always find low-level viremia in anyone on antiretroviral therapy. They never actually develop drug resistance. They just have persistent low-level viremia and there are infected cells, which are largely latently infected cells, which I'm going to explain to you in a bit more detail shortly. So if you have to remember one thing from the lecture, this is sort of says it all, and basically is a snapshot of HIV cure research over the last 3 decades. And there's a lot of interest in this area, not because people are not doing well on treatment. Most people you meet in Australia living with HIV now say HIV is not a big part of their everyday lives. They take a tablet every day, much like people with hypertension might do. However, stigma and discrimination still exists, even in a country like Australia. And to keep the number of people we need on treatment lifelong, and eventually that's 37 million people currently living with HIV, this costs global funders about $30 billion a year. So the drive for finding a cure is really around a global response to HIV, a sustainable long-term response. And this was the front cover of Nature Medicine a few years ago when the IAS, International AIDS Society, developed a global scientific strategy of what we would do to ultimately lead to a cure for HIV. So we first discovered that HIV couldn't be cured in 1997. This was a year after these spectacular new antiretrovirals came available. And we discovered that those antivirals couldn't cure HIV because of the identification of some long-lived latently infected cells, which is now thought to be the major barrier to cure. So that was 1997. Actually, it was the first year latency was discovered in about December in October 1997 by several groups simultaneously, and I arrived in New York December 1997 and was put to working on latency, or working out ways that we could measure and quantify low levels of virus in people that had an undetectable viral load. So it was in New York City at Rockefeller University, and really was the beginning of my interest in this area. I have to just put it into context that in 1996, when these new treatments became available, up until then, pretty much everyone with HIV died. And so when those treatments became available, there were these spectacular responses of people literally getting off their deathbeds, viral load rapidly disappearing from blood. And for the first 6 to 9 months, it was thought antiviral therapy could cure HIV. But it was the discovery of long-lived forms of virus, or latently infected forms, that proved that this was wrong and people needed lifelong treatment. Then in 2009 was the report of the Berlin patient. This man's name is Timothy Brown. He's was the first person cured of HIV, and he was cured through receiving a bone marrow transplant from a donor who is naturally resistant to HIV. So HIV uses a CD4 to enter a cell, but it also uses a chemokine co-receptor. About 95% of strains of HIV use the CCR5 co-receptor. About 1% of Caucasians have a delta 32 base pair deletion in CCR5, are homozygous for that deletion, and don't express CCR5 on the surface of their cells, and are actually completely well. So Timothy Brown had HIV, he was on antiretroviral therapy, was living in Berlin, he's an American man, and developed acute myeloid leukemia and needed a transplant to cure his leukemia. And at the time, his very clever doctor, Gero Hutter, thought, why don't I try and find a CCR5 negative donor for Timothy to receive as a strategy to stop HIV coming back. And that's exactly what happened. And actually, in this case, Timothy Brown stopped his antiretroviral therapy the day after transplantation, and he's been HIV-free for the last 10 years. In 2010, there were several reports of some individuals who can control HIV after stopping antiretroviral therapy. And this is a situation that we refer to as post-treatment control. I started on my first slide telling you that almost everyone rebounds after stopping antiretroviral therapy, but it's almost everyone. There's about a few percent, we're actually not sure how common it is, that people can stop treatment and keep virus under control. This is now called post-treatment control or remission, and it's been an important description or advance because it's coloured a little bit about the goals of cure research. Are we trying to get complete cure, like Timothy Brown, no virus detected anywhere? Or would it be acceptable to just allow people to stop antiretroviral therapy? Their viral load stays at undetectable levels without drugs, and this could also be a great advance, and we commonly call that remission. Just very briefly, post-treatment control is usually described in people who start treatment early after infection, so within about 6 months of infection, usually on treatment for about 3 years, and there have been many case reports, and these are the different cohorts that have been described. So post-treatment control occurs, it's rare, we don't yet know the reason for why some individuals are able to control virus. And in 2014 until now, there's been lots of case reports of remission, or people that have been able to stop antiretroviral therapy. The viral stays undetectable for months, even years, and then suddenly rebounds at a late stage, or what's called delayed viral rebound. So together, although these cases are rare of post-treatment control and delayed viral rebound, these have also influenced the thinking of what we're trying to achieve when going towards a cure. So just where are we at now in 2019? There have been a number of single interventions to eliminate latent virus. None of those have worked, and I'm going to tell you a bit about some of those we've been involved with. We have cured a few monkeys, and many of you, I hope, read the paper by Doherty et al. From Dan Baruch's group using TLR7 agonists and broadly neutralising antibodies. It was the paper that I thought was the most significant advance in the last year, because it was the first demonstration that cure was possible in an animal model. The London patient was just reported a few weeks ago, and some of you may have read about that, so I'll speak briefly about that. So we have a second person cured of HIV, and I think the field very much feels this is not going to be easy. Finding a vaccine has been tremendously difficult for HIV, despite decades of investment. Cure research is at a much earlier phase, but this is complex. The virus has lots and lots of tricks up its sleeve, and many people think, you know what, it's just easier to stay on antiretroviral therapy. It's not my own personal view, because I'm just not that optimistic we can really maintain antiretroviral therapy for everyone lifelong. So just in case you had read about the second man cured of HIV, this was published in Nature about a month ago. He too was a man living with HIV, this time in London. He received a bone marrow transplant from a donor who was homozygous for the Delta 32 mutation. He had a different underlying malignancy. He had Hodgkin's disease. He stayed on antiretroviral therapy for about 18 months after the transplant, and then stopped antiretroviral therapy now 20 months ago. So he hasn't been HIV-free for as long as Timothy Brown, but most people think this man is also potentially cured. And I love this quote from Timothy Brown in a nice article in Science, where he said, "It's great that I finally have someone added to my family. It's been too long." And lots of people do ask me, well, what's happened between 2009 and 2019? Why did it take 10 years to repeat this? And just very briefly, this is because what you're doing here cannot be done to people that just have HIV and are on antiretroviral therapy. The only way you can justify a allogeneic bone marrow transplant in this setting is if the person has an underlying malignancy and needs it for treatment of the malignancy. So first of all, they've got to have HIV. Second, they've got to have malignancy. Third, they have to have an indication for a transplant. And as many of you know, when you transplant bone marrow, you need an HLA match first. And then once you've got your HLA match, you then need to look for a donor who's CCR5 negative, which will only be 1% of all the people that have an HLA match. So very hard to find this setting. However, it has been attempted. There are 10 other cases reported in the literature, and 8 of those cases have all died within 12 months of the transplant because of the complications of either the malignancy returning or infection. And there are 3 others now alive, and one of those people have stopped antiretroviral therapy 3 months ago, and the virus has not yet rebounded. So very exciting area of research, not something we can do clinically, but it has driven lots of new avenues for research towards a cure. And one of those, of course, is gene therapy for CCR5. So to delete CCR5 using either zinc finger nucleases or CRISPR-Cas9, and those clinical trials are happening. So this is a summary of the different clinical strategies that are being evaluated for cure. One is to eliminate whatever virus is left, and one way is to wake that latently infected cell up or put it permanently to sleep. Immunotherapy, anti-PD-1 and other approaches, vaccines and antibodies, and gene therapy. And I'm going to speak to you a bit about 3 major areas of focus in our group: understanding viral persistence on antiretroviral therapy and the role of clonal expansion of infected cells, some background on what we're doing currently in relation to latency reversal as a strategy for virus elimination, and then finally talk about some recent studies we've been doing looking at anti-PD-1. So virus persistence on antiretroviral therapy, a little bit of virology background for you to understand why and what we're dealing with when we talk about latency. So for the non-virologists in the room, HIV typically infects activated CD4 T cells. Once the virus gets inside an activated CD4 T cell, it's the ideal setting for it to undergo productive infection. And we can measure these cells because they all have integrated DNA or detectable intracellular DNA. They generally have intracellular detectable RNA. They express proteins on the surface of the cell. And usually by virtue of lots of virions pouring out of an activated infected cell, the cell will die. Now, in contrast, latent infection is a very different state, common to many retroviruses, common to many DNA viruses, CMV, EBV, all have latent states. And in HIV, what that means is that the virus is integrated into the host genome and is effectively silenced. It doesn't complete the viral life cycle. So it doesn't undergo transcription, or maybe minimal amounts of transcription, doesn't produce viral proteins. And because of that reason, it's essentially invisible to an HIV-specific T cell response or antibody response. And antivirals will also not work because the virus is already integrated into the host genome. And this usually happens in a resting CD4 T cell. But if that cell is activated for whatever reason, responding to a foreign antigen or in some other setting activated, it will start to produce and move into a productive phase. And actually, we used to think of this pretty much as all or nothing, productive or latent. But what I'm going to show you is actually there's probably a spectrum of activity between a truly silenced infected cell and an activated, productively infected cell. So how do these infected cells survive, latently infected cells survive on ART? Well, one mechanism is that the virus infects long-lived cells. So it will infect central memory and naive T cells, which, as many of you will know in this room, have very long half-lives because that's our immunological memory. And we used to think of latent infection pretty much as stable, integrated HIV infection of a long-lived cell, like a naive or central memory T cell. But we now know that these cells can also undergo a level of T cell proliferation and copy that virus into its daughter cells. So there's an element of low-grade background proliferation. And together, this is what really is the major barrier to cure these latently infected cells persisting in someone on antiretroviral therapy. And as soon as you don't notice them when you're on treatment, because then if they're activated and release virus at any time in someone on treatment, that virus can't go on to infect new cells. But as soon as you stop treatment, any virus that reappears or bubbles out of these latently infected cells will go on to set the whole thing off again. We also know that, in fact, on treatment, it's not just latency, there's also what we call an active reservoir. So this is residual virus production from an infected cell that can't go on to infect new cells because someone's on antiretroviral therapy. But these cells can survive preferentially, and we know this happens in certain anatomical sites. Some very interesting work on these cells being largely located in B cell follicles, so with an immunological sanctuary where CD8 T cells can't enter into the B cell follicles in the lymph node. So you have this low-grade residual virus production. It's not viral replication, it's cells that are constantly dribbling out virus in certain anatomical sites. So several years ago, it was shown by some nice papers in Science that the clonal expansion was proven by studying integration sites in HIV. So if you looked, and the classic teaching in HIV infection is that HIV goes anywhere in the genome. If you look at integration sites, and people have looked at this for many years, HIV randomly integrates in many different genes. But if you look in people on antiretroviral therapy, you find cells that have the identical integration site, and that's consistent with clonal expansion of these cells. Because remember, you've stopped any new rounds of infection, so the only way that these identical integration sites could have occurred is through clonal expansion. And there's a lot of interest in these clonally expanded cells. They can account for almost 50% of infected cells. The site of integration may potentially give the infected cell a survival advantage, or potentially these cells are proliferating and expanding because they're antigen specific. We don't yet know the answer to that. But we have been very interested in the site of integration because maybe that is giving this infected cell a means of surviving, which is more which is different to if the virus is in another part of the genome. And I'm going to talk a little bit about some work from a Dutch postdoc with me, Jori Simons, who's been developing a high throughput assay for integration site analysis. And just very briefly how this assay works is that Jori uses enzyme-mediated and mechanical disruption to dissect or cut up the host genome. And here you can see the host genome with HIV sitting embedded inside it. And then those areas where you've got HIV virus and host virus are tagged with an N linker. And when you find the identical integration site, but from with different lengths of the genome, you know that that virus has come from a different cell. He then uses a really nice strategy for amplification with one primer sitting in the linker and the other primer sitting within the HIV genome. He gets a biotennylated product to purify these basically mini integration sites, and then uses streptavidin and capture a series of washes, a second round PCR with a barcoded PCR product, and then can examine those using a deep sequencing library to see where the integration site is in which human genome. And this is just shown again here in a nice diagram, which eventually you can then do a search to find where your clonally expanded or single integration sites are. And what Jori has found, using his own analysis, as well as all the published work on integration sites in the literature currently, is that if you look at what how many integration events you'd expect in certain genes, so the expected p-value, because obviously a big gene will have a lot more chance of more integration sites compared to a small gene, compared to what you actually observe, you can see there's enrichment for certain genes where there's overrepresentation of HIV in clonally expanded, in expanded clones. And some of these genes may be familiar to people in the audience, so I'd love if any of you are working on these. Batch 2, MKL2, STAT5B are repeatedly described from many different cohorts, showing that HIV in those sites is preferentially expands. And so Yori's been doing some really nice work looking at which genes are more common in cells that are clonally expanded compared to unique integration sites. And this just lists some of these comparisons between single sites and clonal sites, and you can see they really are very different. There's a whole lot of genes that are far more commonly represented in clonally expanded cells. Now, when you actually look across the spectrum of different T cells, and again, at WEHI, I'm sure I don't need to explain the pathway for T cell differentiation. It's much more complex than this. I know it's not entirely linear, but many of you, of course, know that there's naive T cells, stem cell memory T cells, central memory, transitional memory, effector memory, and terminally differentiated cells. And actually, if you look in people on antiretroviral therapy, you can find integrated virus in all of these different subsets of cells. And we've had this very nice collaboration with UCSF, where people with HIV on antiretroviral therapy undergo labelling. They get infused with deuterated heavy water infusions to then label and look at turnover of these cell subsets. And those cell subsets are sent to us, where Yori looks at the percentage of, where the integration sites are and the percentage of clonal expansion. And you can see here very clearly that we see far more expanded clones in these differentiated cells, effector memory and terminally differentiated. And also, interestingly, you can see the same integration site within multiple cell subsets, arguing or potentially saying that the HIV integration site the infection can persist through T cell differentiation. So here's an example of the size of the diamond tells you the frequency of that integration site detected. You can see here large clonal expansions in these terminally differentiated cells, but the same integration site is seen in an effector memory as well as a central memory T cell. So this tool is giving us lots of fundamental insights into how the virus can persist in certain sites, and hopefully one day the really important question is why in some sites is there clonal proliferation, and can we reverse that in some way? And just a very interesting paper recently that managed to combine integration site analysis with full-length viral analysis. I haven't spoken about this at all, but another very important concept in HIV is that most of the integrated proviruses that persist on antiretroviral therapy are defective. They've got large deletions or stop codons, and therefore the virus can't replicate. You actually need an intact genome to give you a replication-competent virus. And there are some really elegant ways now to detect this intact virus. Of course, full-length sequencing, laborious and expensive, but some new approaches using droplet digital PCR just recently published. And this new technique, done on very limited numbers of patients, actually looked at the integration sites for intact virus versus defective virus, and were actually able to show that the intact virus was preferentially found in genes that were more likely to be silenced. So they were longer distance away from transcriptional start sites. So where the virus lands and persists may give us some clues in how it can persist for so long on people on treatment. So a very quick summary of this part of the work. HIV persists on treatment in multiple forms. Clonal proliferation plays an important role. We think the integration sites of expanded clones have multiple distinctive features. They are enriched in genes associated with cell survival. They're more common in effector memory and terminally differentiated cells. And identical sites in multiple subsets is consistent with differentiation of an infected cell. And future work we're focusing on is to understand why and how these specific genes are targeted, and we're trying to do that using CRISPR-Cas9 models to introduce an LTR reporter virus into these specific genes. So I'm going to switch tack a little bit and talk now about interventions that we're being tested and investigated to see if we can eliminate latency. And one intervention that I I and my group have been very active in since over the last decade really is to see whether we could reverse the state of HIV latency. This is often called a shock and kill approach. And so the idea of this is that here is your latently infected cell, integrated provirus, minimal transcription, no protein expression, invisible to any T cell or B cell response. And the idea is to activate that virus using a latency reversing agent, or what we commonly call LRAs, to increase the antigen exposure or virion release from the cell. And this will either lead to viral-induced cytopathic death, because normally when virions are released from the cell surface, it induces cell death. Or alternatively, this allows for immune-mediated killing because this previously hidden provirus now has antigen exposure. And this is all done in someone that's on antiretroviral therapy. So any new virus we might generate by this latency reversing agent can't go on to infect any uninfected cells. And there are lots and lots of latency reversing agents that have been demonstrated to work in vitro. This is a nice review done by a PhD student in my group, Yuri Kim, and it just shows you the different classes of drugs which can all activate latency. So up here you can see there are epigenetic modifiers, so HIV transcription is regulated by acetylation and methylation just like any other gene. There are drugs such as TLR agonists, which I'll talk a little bit more about how they might work. Protein kinase C agonists, which activate transcription factors needed to drive HIV transcription. Here we've got SMAC mimetics. We're just having a chat about SMAC mimetics as non-canonical NF-kappa B activators. And so there's many, many of these agents. And they work largely, as I mentioned, by epigenetic modification, so altering the transcription of the integrated provirus, very similar in concept to how other genes are regulated, or by activating transcription factors such as NF-kappa B, which are important drivers of transcription. Now, when you put these drugs into people to try and measure latency reversal, so we evaluate these drugs in vitro using cell line models, ex vivo with taking cells from patients on stable antiretroviral therapy. And also some of these drugs have been put into people to see whether you can reverse latency or deplete latently infected cells. And this is, once we go into people, it's actually quite hard to measure latency reversal for a few reasons. First of all, these infected cells are very rare. There's about 1 in a million cells are infected with latent virus. Most of them are sitting in tissue, not circulating, and so they're very rare, so you need good sensitive assays. And we're really just looking for virus production, meaning that if you look, we can look inside a cell, so you've got integrated virus, and if there's an increase in cell-associated RNA, it means you've activated transcription. And if you're lucky, maybe you also can get protein expression, hard to measure, and you might produce virus in the plasma, and you can detect that looking at RNA in plasma or HIV virions. Tricky thing to measure, because if this cell is now suddenly exposed to immune-mediated clearance, you may kill this cell before you can actually measure virions in plasma. But most of the studies look at the number of infected cells by measuring HIV DNA or integrated DNA, how much transcription there is by looking at either unspliced or multiply spliced RNA, and then looking at virions in plasma by measuring plasma-associated RNA. So we've been active in this area for a long time. We performed one of the first clinical trials of HDAC inhibitors published back in 2014, so a very long time ago. And what we did show that when you gave an HDAC inhibitor, varinostat, a very modest, not very potent HDAC inhibitor, you did actually increase the cell-associated RNA, so you could activate transcription. But one of the big problems with HDAC inhibitors are that there's a whole lot of off-target effects. And this is just showing a gene array map of individuals enrolled in the study with blood collected at baseline in green or at day 84, so about 3 months after stopping vorinostat, and you can still see multiple altered genes. So not great for people that are otherwise living on taking 1 tablet a day with a normal life expectancy. So we need better ways of activating virus that has minimal off-target effects. We've also become aware that there are actually multiple steps needed to reverse latency. It doesn't just go from latent infection all the way through to productive infection. You can actually just activate transcription, where you can detect the initial steps of transcription, unspliced RNA, or you can activate translation, where you now need multiply spliced RNA and protein expression, or you can activate productive infection. And that in order to reverse latency, we may need to look across this whole spectrum, and HDAC inhibitors really pretty much just did this first step of activating initial steps of transcription. And the other thing was that we found that the number of infected cells did not decrease. So these drugs could shock, but didn't seem to kill. And this is one of the trials we did with the vorinostat, and another trial with another HDAC inhibitor, panobinostat, done by colleagues in Denmark. And we now know, this is not work done by us, but we now know that why HDAC inhibitors don't seem to work so well. And this is a little bit complicated for the non-virologists here, but these are basically the steps needed in order to produce spliced RNA, which is required to make virions. And if you fully activate a T cell with anti-CD3, CD28, you progressively move through all of these steps, efficiently make spliced RNA, which then drives virion production. But if you activate the cell with an HDAC inhibitor, and here this was used romidepsin, you can see here that these first steps of transcription are initiated, meaning that you initiate transcription and you elongate it, but you don't progress to these stages of splicing. So there are blocks from HDAC inhibitors. So either we won't use them, they're not safe enough, they don't produce multiply spliced RNA, or perhaps they can be used in combination. Now, many of these other interventions have also undergone clinical trial evaluation, and most of the studies have shown us that you can you can reverse latency in vivo, but we do need more potent, more specific and less toxic LRAs, and probably latency reversal alone is not going to eliminate latent infected cell. You need a second intervention, or what we call a kill intervention. And we've been together, this is really work led by Damien Purcell from the Doherty, but a really nice collaboration through a development grant with Brad Sleeves here from WeHi, trying to discover new and more potent and more specific latency reversing agents. And the approach that we're using is a really elegant cell line that Damien developed that has one reporter under the control of the viral LTR, shown here in red, and another reporter under an unrelated promoter, here CMV, shown here in green. And in the drug screening that Damien did together with Brad, they identified drugs that had preferential activation of the HIV LTR over the CMV promoter. And we did this in this cell line that Damien's lab made. And then we've enrolled a large number of patients. This is work done at the Alfred. These are HIV-infected individuals on antiretroviral therapy that donate cells through leukapheresis. It's a really powerful tool that we now have. Patients love it because it's a great way to participate in a trial, and it's just a one-off 4 hours of your time. I mean, you're hooked up to lots of machines. It's definitely an intervention, but it's safe, and it's a one-off contribution. And we then get billions of cells from people that we can store and go back to and test. And we use those cells to test for activity in a patient-derived model. And what Damien's group has found is a sort of a novel class of drug, imidothiazoles, that also activate latency and potentially have greater specificity. And Brad has just published this literally just about a week ago in Journal of Medicinal Chemistry. So all of the latency reversing agents I've talked about so far largely focus on activating transcription. So we're now beginning to call them transcription activating LRAs. But the latency reversing agents that seem to have a bit more promise are these drugs that we now call immunomodulatory LRAs. So they're not just acting on the infected cell, they also have enhanced immunological effects which could potentiate clearance of this infected cell. And some of the drugs that fall into this group include immune checkpoint blockers, which I'll talk about briefly. IL-15 does this, and also the TLR agonists do this. And this was the paper that I asked the students to read. This was an antibody, broadly neutralising antibody and a TLR7 agonist that was given to monkeys that were infected with something called HIV, which is a hybrid virus between a monkey virus, SIV, with an HIV envelope inserted into the monkey virus. And what they found was that these monkeys essentially cleared infected cells, and the thinking is that the TLR7 agonist worked by activating the CD4 T cell, essentially latency reversal, allowing for expression of viral proteins. And the antibody, here the antibody used was PG121, was then able to recognise this protein and activate NK cells for clearance. And the TLR7 agonist probably had a dual effect. It was very hard to tease this out in the monkey model, but based on previous in vitro work, it activated the virus and also activated NK cells. And for those of you that have read the paper, this was the main finding, was that when they infected monkeys, put them on antiretroviral therapy, then gave the intervention and then stopped and looked for time to viral rebound, or whether the monkeys controlled virus by themselves. And you can see here in the sham or placebo monkeys, all the monkeys rebounded and then eventually controlled virus, but at a detectable level. In contrast, the monkeys that received the antibody PGT121 and the TLR7 agonist, half of them appeared to be cured, meaning half of them didn't rebound. So a lot of excitement with this paper. This was HIV, which is not a very pathogenic virus. The monkeys were treated very early after infection, so maybe they had tiny reservoirs. There was a high frequency of controllers, and also, was the TLR7 agonist doing much? Because even the antibody alone managed to cure a few monkeys. And TLR7 agonists are being developed by Gilead, actually, as latency reversing agents, and currently in human studies. Now, I'm conscious I'm running a little bit out of time, so I'm just going to quickly show you a few results on the work that we're doing with anti-PD-1. So all of you, I know, will know lots about all the different immune checkpoint markers and their ligands, and that we now have antibodies that effectively block this interaction. Just like in malignancy, there's been a very high interest in immune checkpoint markers in HIV infection, because PD-1 and CTLA-4, many of the other markers are all upregulated in CD8 T cells in untreated HIV infection, and actually still remain elevated in treated HIV infection. As you know, there are antibodies that have been really successful in regenerating or reinvigorating immune responses in malignancy, so could we do this in HIV? They may have a role in cure, because these reinvigorated HIV-specific T cells could be important for clearing latently infected cells. But one key difference in HIV is that your target also expresses PD-1. So latent virus is actually enriched in PD-1 positive cells in blood and lymph node, and also in CTLA-4 positive cells. So we've been interested to see whether anti-PD-1 could potentially reverse HIV latency, and the idea here is that latently infected cells express PD-1. The antibody effectively takes the brakes of PD-1 off the virus, and this drives productive infection, leading to cell death. So can these drugs act as latency reversal agents? And of course, at the same time, they should enhance T cell killing. But we've just been primarily interested in this latency reversal component. And together with collaborators Nicolas Chamont and Remy Fromentin, we were involved in some really nice studies showing that anti-PD-1 does effectively reverse latency. Here, this was shown by adding the ligand PD-L1, so that if you occupy or bind to PD-1, you limit the virus production. And then ex vivo, if you take patient cells and you stimulate them ex vivo with a stimulus such as SEA, SEB, and add anti-PD-1, you can potentiate the amount of virus released in the supernatant. And the same response can also occur with other latency reversing agents, such as bryostatin. And you can see here MOC or isotype control, minimal virus production, and then when you add in anti-PD-1, you substantially enhance virus production. I'm just going to quickly skip through these slides on in vitro work. When you look at people that receive multiple doses of anti-PD-1, there's been a few case reports. And just, this is a bit complicated here, this is a case report from a French group. When they looked at HIV DNA, which is the number of infected cells in blue, you can see this steady decline in infected cells after repeated administration of anti-PD-1, here being pembrolizumab. And in the same paper with Nicolas and Remy, another patient from Montreal also showed this decline in HIV DNA with repeated administration, potentially arguing that, that you're getting clearance of infected cells with prolonged administration. Now, in HIV, we'll never be able to give anti-PD-1 at repeated doses, largely because of its significant toxicity. But maybe we can give anti-PD-1 at single or perhaps 2 doses. And we've recently been involved in a prospective clinical trial evaluating pembrolizumab in individuals with HIV and advanced cancers, and looked at what happened after the first dose of pembrolizumab. And you can see here where we measured cell-associated or unspliced RNA. You see a significant increase by a week after infection. We also saw an increase, but this didn't reach statistical significance, in plasma viremia. And no change on total HIV DNA. Had this interesting early decline, which I suspect is redistribution of PD-1 positive cells. And overall, this is a sort of marker of transcription and increase in cell-associated RNA to DNA ratio. And we've got lots of other very interesting studies, because here, as opposed to cancer, we know the exact antigen. So a lot of work on using cytoph and transcriptomics to understand those very initial early responses to HIV following anti-PD-1 in vivo. What the field is really waiting for are clinical trials in HIV-infected people not with cancer. That's because there's so many other variables that are occurring in these individuals receiving anti-PD-1. And there are 2 studies currently underway at the NIH giving a single dose of anti-PD-1, pembrolizumab, and another with one of these newer anti-PD-1 drugs, cemiplimab. So just finally, in summary, complete elimination of virus on ART is a big challenge, largely due to these variable forms of latency and clonal expansion of infected cells. HIV cure in the human setting has been really rare. It's only so far happened following stem cell transplantation. But in monkeys, we've got this recent new exciting evidence that perhaps TLR7 agonists and broadly neutralising antibodies may play a role. Latency reversal, I think, is going to be an important part of cure strategies, but won't be work on its own. It's there to expose these latently infected cells. And we've shown evidence now of anti-PD-1 reversing HIV latency in vivo, as well as ex vivo and in vitro, and may also enhance HIV-specific immunity. We haven't yet examined that in great detail, and further studies in the absence of malignancies needed. And before I end, I just thought I'd highlight that there's a lot of excitement about what we can do with HIV with the tools we have. So I'm a big believer that we still need an HIV vaccine and a cure, but we do know that if we test people more, And people know that when they're HIV positive, if they're treated early and they stay safe with a whole range of other ways we have available to prevent HIV, particularly pre-exposure prophylaxis, we can probably end HIV in a country like Australia. But I think with these interventions alone, it would be very difficult to do that globally. And these are just a number of my group that have played a very active role in many of the studies that I've shown you. We collaborate very widely, both across Australia and internationally, and my funding support. Thank you very much.
Plenty of time for questions.
I had a question with the Berlin patient and approaches like gene therapy. Is there any consequences with not having CCR5?
Surprisingly not. People, as I mentioned earlier, about 1% of Caucasians are CCR5 delta 32 homozygous, more common in Northern Europeans and Southern Europeans, and no normal healthy lives, except for West Nile virus, has been reported to be more severe in people that are delta 32 homozygous, but otherwise it's compatible with normal immune system. Should just say though, and I didn't get into this detail, that HIV can use a different co-receptor to enter cells. It's predominantly used as CCR5, but it can also use CXCR4. Most circulating strains of virus are CCR5 using, but you can also be infected rarely with an X4 using virus. And there's reports, when CCR5 became a very sort of exciting finding in HIV back in the mid-90s, there were reports of people with the delta 32 homozygous mutation acquiring CXCR4 HIV. So it's not absolute.
Have you heard of any of these new LRAs being used to treat other latent infections?
Yeah, it's a very interesting question because depending on which class of drug you're looking at, they may or may not have activity against other retroviruses or even other DNA viruses. And one group of drugs, certainly the histone deacetylase inhibitors that I spoke to you quite a bit about, they do activate the LTR from endogenous retroviruses, from some DNA viruses, HBV and CMV. So actually, the reports are usually a complication of those drugs leading to reactivation of other viruses. They're not really being exploited for virus elimination.
You have those 4 stages going from a latent cell to a productive infectious virus. Sorry. Does it happen the same the other way when the virus goes from being productively infectious to latent? And can you detect like the translational active stage that's not sort of productively infectious?
Yeah, that's an excellent question because I didn't discuss at all about how latency is established. So that's a whole nother story and one also that we've been really interested in for a long time. And there's 2 main theories of how latency is established. One is that you can infect very inefficiently, but you can directly infect resting cells. The virus integrates and basically gets stuck there because it hasn't got all the transcription factors to complete the viral life cycle. And the other is that you have an activated infected cell that survives that. Just like in a effector memory response, you have all these activated antigen-specific effector memory, and some of them survive and revert to central memory. So the sort of the dominant theory about establishing latency is survival, exactly what you're saying, going back down the other way. So it survives because the virus is in a site that favours transcriptional silencing, or it's just random, that in any effector memory response, you'll eventually lay down central memory and carry the virus with them. So it's quite a hard thing to study in vivo, but in vitro you can establish latency both ways.
So in the Lewin patient and the London patient, do you think the
Yeah. Yeah, no, another great question. I think in these two men, there are two things happening. One is that you do reduce the reservoir through transplantation, and there have been many other reports of transplantation from a donor who was wild type for CCR5. In fact, we've reported a case that we looked after at the Alfred, and the reservoir decreases after transplantation. And that probably happens with graft versus host disease. So it's probably not the conditioning treatment. First of all, the conditioning treatment for the Berlin and London patients were very different, and there's been a whole host of reports of whether people had total body irradiation or anti-CD52 or OKT3, all these different conditioning treatments don't seem to be linked with it, but graft versus host disease seems to be important. So just like I'm sure many of you know about sort of graft versus leukemia, you probably have the same thing. You're wiping out someone's bone marrow, any residual recipient cells, which some of them will be infected, are also cleared both by the conditioning and graft versus host disease. So you've got graft versus host disease that reduces the reservoir. And in some cases of donors that were wild type to CCR5, there's been 3 reported cases of then those people had no detectable reservoir, so their doctors in the setting of a clinical trial stopped antiretroviral therapy, and they all had very delayed rebounds. So it took sort of months, 8 to 10 months for the virus to rebound. So you have reduced the reservoir, but then you've got no immunity because you've got a totally new bone marrow that's never seen HIV. So in the London and Berlin patients, I think it's the graft versus host disease that eliminates the reservoir. And any virus, because you only need 1 or 2, in fact, you get infected from HIV with a single virion that crosses the mucosal membrane. So if you have any 1 or 2 virions left, they couldn't take off again in those 2 patients because they've got no target cells. So one problem with gene therapy at the moment, and most of the published work is on ex vivo gene therapy. There's some in vivo gene therapy in monkey models, but in humans it's ex vivo. So blood's taken out, modified and then reinfused, or stem cells are taken out, modified, reinfused. You can get up to about 5 to 15% of gene-modified cells. But when you stop treatment, the virus just rebounds again. So in the animal models, there was a selective advantage for the CCR5 negative cells, but that hasn't played out in humans, and maybe you just need higher levels of these modified cells.
Any last questions?
Quick question. So is there a correlation between the genes that this virus incorporated into and RNA half-life, and also having been that like some patient inhibitors have been shown to cause RNA degradation, so I'm wondering if you've looked at the degradation of the viral RNA.
You mean RNA half-life of the virus?
No, of the genes. So do they prefer to incorporate themselves into genes with a longer RNA half-life?
Yeah, I don't know if that's been looked at. It's certainly, HIV is more commonly found in actively transcribing genes. And there's one theory that it's more common in those sites because at the time of infection, that chromatin's more exposed, and so therefore, the chromatin that's more exposed with its active gene expression at the time of infection favours virus persistence. I actually don't know how you measure RNA half-life. Not thought about that, but people have looked at whether there's expression of those genes either at the time the cell was infected or when you see it latently. But I'd be interested to hear more how you measure RNA half-life. And the second question was degradation. Again, when you measure infected cells in people on treatment, you can measure DNA, which is integrated and stable, and you can measure transcripts that it's very hard to know how stable they are intracellularly. If it enhanced degradation, I would have thought you'd reduce the amount of intracellular transcripts, but actually post-panobinostat you increased intracellular transcripts. But the degradation is an important thing to measure because obviously there's a steady state and it's not, as you're producing RNA, it's being degraded. So we know that that's actually quite stable, the amount of cell-associated RNA. But I don't think we have good tools to measure turnover of intracellular RNA.
So based on, for the interest of time, we'll stop it here. But please, students, come to boardroom. We have a lunch there to ask further questions. Also, our guests from PDI are more than, excuse me, welcome to come. Sharon donated kindly a bit more time of her precious time to join us there. And please join me and thank Sharon for a very fascinating talk and journey through HIV latency and current therapeutic approaches. Thank you.